Hello guys and welcome back to Excelsior Academy. So in today's lecture, we are going to talk about the lipids which is present in the mitochondria. Okay. So before that, we have talked about the what all proteins are there, how proteins are getting sorted. So if you remember, what is the signal sequence over here? Yes, anyone who remembers? So as I said, in the case of nucleus, it is NLS. In the case of ER, it is KDEL or KKX. This is for soluble protein. This is for your membrane bound protein, right? In the case of lysosome, it is mannose 6 phosphate. So in the case of mitochondria, it is known as pre-sequences. Okay. Now let's talk about the lipids which is present in the mitochondria. So not only the proteins, but also the lipids of mitochondrial membranes are important for from the cytosol. In animal cells, sphingolipids, cholesterol, phosphatidylcholine, inositol, serine, they are all synthesized in the ER and then transported to the mitochondria. Now, how these are transported? If you remember, in the ER, when the synthesis happens, the vesicle which forms. So this vesicle will be carrying all the cholesterol, choline, serine, inositol. Okay. Now this will go to the mitochondria and it will fuse over here. Okay. So now we have seen like how the vesicles are formed with the help of your COP1, 2 and clathrin, right? With the help of these code proteins. And then we have seen how it gets fused. So fuse means they're where we have talked about the V snare and T snare. So there the fusion happens. So this is how the important lipids are transported to the mitochondria or any other organelles. Okay. Now mitochondria also catalyze the synthesis of unusual phospholipid, which is known as the cardiolipin. Now cardiolipin, it contains 4 fatty acid chains. I'll show you the structure. Yeah. So this is the structure of your cardiolipin. And what do we see here is that they have got the ether bond instead of, see, in our normal phospholipids or the fatty acids, you have got the ester bond. Okay. Now here it is the ether bond, and that makes it more stable. Okay, more strong and stable. Now ये कहाँ पे present है? They are only found in the inner mitochondrial membrane. Okay, they are present only in the inner mitochondrial membrane. Now can anyone tell me ऐसा क्यों है? Any guesses? So as I already said that it is present in the inner mitochondria. What is the speciality of inner mitochondrial membrane?
It is less permeable to molecules and ions.
Okay. Then what else? Yes. So inner mitochondrial membrane, as we know, they form the cristae, right? To increase the surface area. Now, in order to maintain that structure, कुछ ऐसा रहता है, right? And then you have got this outer membrane. So to maintain the structure of your inner mitochondrial membrane, cardiolipin plays very important role in that. And of course, So it helps, as she said, like it is mainly impermeable to the outside of many molecules, right? So it is because of this cardiolipin, it is maintaining the more amount of your proteins. जैसे कि हमें मालूम है यहाँ पे आपके complex 1, 3, 4, 5 and their copies will be, so many copies will be present. Okay. Now this cardiolipin makes or ensures that these proteins, these integral proteins are remain intact. Usually क्या होता है कि आपके phospholipids are dynamic in nature, right? When I say dynamic in nature, it means what? On the membrane, phospholipids will be keep on moving here and there, right? They laterally move or transverse movement is there. Now, with the help of the cardiolipin, that's not possible in the case of the in the mitochondrial membrane. अगर मैं बोल रही हूँ यहाँ पे आपका कॉम्प्लेक्स 1 है, 3 है, 4 है, 5 है, तो वो यहाँ ही है. ओके, इट्स नॉट लाइक ये मूव होता हुआ यहाँ चला गया या यहाँ चला गया, which happens normally in the case of the plasma membrane. Did you get the point?
Yes, ma'am.
Okay. Good. Okay, so they are localized in the inner membrane of mitochondria. It improves the efficiency of the oxidative phosphorylation. It also restricts the proton flow across the membrane. The transfer of lipids between the ER and mitochondria. Now, this is, guys, really very important. Okay, please concentrate this one. So, the transfer of lipids between the ER and the mitochondria takes place at sites of close contact between the ER and mitochondrial membranes and is thought to be mediated by phospholipid transfer proteins. Okay, which extract the single phospholipid molecules from the membrane of the ER. So what is the meaning of this? कि it takes place at the sites of close contact between the ER and mitochondria. इसका क्या मतलब हो रहा है? See, ये आपका हो गया पूरा सेल, ठीक है? Let's say here you have got the nucleus. Now nucleus के आजू बाजू में you have got your ER. Okay, now you have got many mitochondria present. So we have got in eukaryotic cell around 2000 mitochondria are present. Okay. Now, whatever site, okay, whatever part which is in near contact to the ER, there this transfer is gonna happen. Okay. So first of all, your mitochondria are not restricted to one site. Okay, as I mentioned before also, on the cytoskeleton, on your actin and microtubules filaments, what happens is that they keep on move. Okay, so kaisa hoga? If let's say that this mitochondria is getting increase or the transfer of phospholipid is happening, so the portion of ER which is near or which is like the shortest distance between the ER and mitochondria, now here this phospholipid transfer proteins will come and do the transfer. ठीक है, अभी यहाँ पे ये साइड से ये साइड में होगा. What I want to say is that the transfer is not happening यहाँ से और यहाँ से. Okay, so the addition will happen in the nearby contact. ऐसा नहीं कि यहाँ से वेसिकल बनेगा, वो इतना मूव करेगा और यहाँ आके फ्यूज करेगा. Did you get the point? The shortest distance between the ER and the mitochondria, there this phospholipid transfer will happen with the help of the phospholipid transfer proteins. Okay. Did you get this concept?
Yes, ma'am.
Okay. Now the lipids are exchanged between the mitochondrial outer and inner membrane at the sites of contact between them. So that can happen now that so many contacts are there with, there are many places where the translocators are, fused. Now this is your phospholipid transfer protein. So now you can see that here you can see you have got your ER membrane, right? ये आपका ल्यूमन हो जाएगा, तो आपका ER ऐसे present है यहाँ पे. Now phospholipid transfer proteins क्या कर रहा है? It is taking one of the phospholipid and it is transferring to the माइटोकॉन्ड्रियल का आउटर मेम्ब्रेन. ओके, दिस इज योर इंटर मेम्ब्रेन स्पेस. तो यहाँ पे फिर आपका इनर मेम्ब्रेन आ जाएगा. करेक्ट. सो यू कैन सी ओवर हियर कि कैसे एक एक पिंच ऑफ करके वो माइटोकॉन्ड्रिया को ट्रांसफर कर रहा है. एंड दिस इज हाउ द लिपिड इज ट्रांसफर टू द माइटोकॉन्ड्रिया आउटर मेम्ब्रेन. ओके.
Yes, ma'am.
Good. Now transport of metabolites across the inner membrane. So now what we are going to see is the how see, first of all, the mitochondria, we know that it is involved in the generation of your energy. Okay, so we know that many what to say, many metabolites, for example pyruvate का है, pyruvate glycolysis से बनता है, right? So which happens in the cytoplasm. Cytoplasm से अब उसको mitochondria में लाना है, where that too in the matrix of your mitochondria. Correct, because there your Krebs cycle will take place. Now all these are only possible with the help of the transport proteins. Okay. So the function of mitochondria in the generation of metabolic energy requires efficient transport of small molecules into and out of the mitochondria. Let's talk about ATP itself. So ATP कहाँ synthesize होता है? So we know that ATP synthesizes in the mitochondria, correct? If you remember, ये outer membrane हो गया, ये आपका inner हो गया. So on inner, you have got those complex 1, 2, 3, 4, 5 are there. यहाँ पे आपका H plus concentration increase होता है in the intermembrane space. When this proton gradient is formed, the proton is moving from intermembrane space to back to matrix, right? तो ऐसे में क्या होगा, आपका ATP synthesize हुआ है यहाँ पे. Correct? It means ADP plus phosphate was also present in the matrix of the mitochondria. Correct? बट अभी हमें मालूम है जब भी कुछ काम होता है या जब भी ATP हाइड्रोलाइज होता है या यूज होता है, it always happens in the cytoplasm. Okay, so what I want to say is that the substrate for ATP synthesis is present in the cytoplasm and the product is present, the ATP is present in the matrix. अब क्या है, in order to use ATP, this ATP should be present in the cytoplasm and the substrate should be present in the matrix, right? Did you get this thing? Yes, Sanvi, it is clear. Is this part is clear?
Yes, ma'am.
Okay.
Yes, ma'am.
Now for this, so the ATP synthesized within the mitochondria, it has to be exported to the cytosol, right? While ADP and PI, which is needs to be imported from the cytosol so that the ATP synthesis can take place. Now the outer membrane of mitochondria is freely permeable, but many proteins of the inner membrane are like small molecule transporters. Did you get it? So it's not permeable. Inner membrane is not permeable, but it has got many transporters. So if you remember in the previous lecture, we talked about a small molecule transporter known as OXA1, the translocators. Did you remember?
Yes, ma'am.
Yeah, so with the help of this OXA1 on the inner membrane only, you can have your proteins getting integrated. Okay, now the energy which is required to drive the transport of these small molecules into and out of the mitochondria, now this is provided by the electrochemical gradient which is generated by the proton pumping across the inner mitochondrial membrane during the oxidative phosphorylation. So abhi hame pata hai that the proton gradient which is formed during the ATP synthesis, along with the ATP synthesis, it also helps in the movement or the import export of your ATP and ADP from the cytosol to the mitochondria and vice versa. Okay. Now look at the diagram over here. Now, what do we see here? One second. Yeah. So this is your intermembrane space. ये हो गया आपका outer membrane. Okay. Now here the inner membrane. So on the inner membrane, what do we see? So many small molecule transport proteins are present. Okay. Now let's go with the first one. So when this H plus concentration is increased in the intermembrane space, your electrochemical gradient is formed. Okay. The gradient is formed. Electrochemical or this proton gradient, right? Okay. Now your small molecule transporters, see, with the help of this, what is happening? ADP is coming over here with exchange of ATP4. Okay. Now, similarly, phosphate is also coming in exchange of H plus. Then pyruvate is coming in exchange of OH minus. Okay. So all this is helping in the, all this is possible with the help of the proton gradient. Here we are not using ATP. Okay, because this is all meant for the ATP synthesis. अब हम उसको ही बनाने के लिए हम उसको use नहीं कर सकते, right? It doesn't make sense in the nature then. Okay. Now, keeping this diagram in your mind, let's talk about like what it is, how the transfer is happening. Okay, so the proton gradient across the inner mitochondrial membrane, it has both chemical and electric components. Okay, for example, the transport of ATP and ADP across the inner membrane, it is mediated by a integral membrane protein which is known as your adenine nucleotide translocator. Okay, so this is helping in the transport of ADP and ATP. Now, because ATP carries more negative charge than ADP, -4 compared with -3, this exchange is driven by the voltage component of the electrochemical gradient. Okay, is this part clear? We can go back to the figure. See, look at the figure, like how this is helping. Is there any doubt in this topic? Same thing like what we have discussed now. So this proton gradient will help in the exchange of your ATP and ADP. Similarly, we have for the phosphate along with the OH minus. Okay. Same way for your pyruvate also. So the pyruvate is there which is present and it is getting exchanged with the OH minus. So eventually what we are making sure that this proton gradient is present so that later the ATP can be synthesized. अब भी हमने हमेशा क्या पढ़ा ऑक्सीडेटिव फॉस्फोराइलेशन में that there is an intermembrane space वहां पे आपको proton gradient है and that helps in the ATP synthesis with the help of the complex 5. Now this is also happening along with the ATP synthesis. You need the continuous supply of the substrate, then only the ATP will be synthesized. Okay. Guys, clear?
Yes, ma'am.
Okay, so you can go through it. What is written, or these are directly taken from the Cooper only. Any doubts?